webs, right? And our understanding of the food webs comes from observations, field observations, what we have observed, the dynamics of the different components in the sea, and how they interact with each other. So all this could be observations either from lab-based experiments or also from field sampling. So if you look at this slide here, it's mentioned that the bacteria and phytoplankton coupling. The word coupling is when you see two components linked to each other from observations from in a single study. For example, if you're measuring bacterial abundance at a certain site over time, and if you look at the compared it with the chlorophyll A concentration. So chlorophyll A is a proxy for phytoplankton. So we measure chlorophyll A because it's easier to measure, because counting phytoplankton is difficult. So if the coupling holds true, then the relationship should be somewhat linear. Okay, at least a positive relationship where when chlorophyll A concentration increases, bacteria abundance also increase. This should be the case because if you know that if the primary production, if the primary producers increase, that means there are more primary production. Okay, that means there's more contribution to dissolved organic matter, and that means the bacteria will also increase because there's more food for the bacteria. So this is a form of coupling between components in the marine food web. So from these observations, you are able to see the relationship between the bacteria, which is now known as the main heterotrophs, and primary producers, the main phototrophs. So the landmark study that carried out, so they call this kind of analysis meta-analysis, which means you gather the data from different publications. So the first one was Cole et al. In 1988, if I'm not mistaken. I'm not sure about the year. So Cole et al. Publications from lakes, studies from lakes, coastal waters, oceans, and all that. And he found a very nice relationship between bacteria and chlorophyll A. Also between bacterial production versus primary production. Okay, so from this relationship, he further confirmed the coupling between bacteria and phytoplankton. And other relationships that you probably can guess is between bacteria and then your protozoa. Okay, so in most studies, in the earlier studies, they focused on HNFs. HNFs are heterotrophic nanoflagellates. Okay, they specify heterotrophic because nanoflagellates can be mixotrophs, which means some of them have chloroplasts in them and can carry out uh, photosynthesis. So they specify heterotrophic nanoflagellates. So this is also counted under epifluorescence microscope. Same procedure as DAPI, except they use a different fluorochrome. Usually we use primulin. Uh, Nanoflagellates are quite easy to identify because you'll see a pair of flagella, or sometimes you'll see flagella sticking out from the protozoa. Okay, so from your understanding of the microbial food web, you know that protozoa will graze upon bacteria. So what you should see should be this also. Okay, because as the number of bacteria increases, the number of prey for the protozoa increases, you should see a significant coupling also. So in some occasions, for example, you see patterns like this. Okay, more likely. Okay, so this is considered uncoupling. That means in the site that you are studying, bacteria and chlorophyll, there's no the traditional sense of coupling between the two components. Okay. Is this possible? Is this possible? Where the variation in bacteria is so another way you can say this observation is the variation, the variation in bacterial abundance is independent of chlorophyll concentration. So is this possible? Remember, whenever we talk about any component, any nutrients that we were studying, whenever we talk about a component, we always talk about the input and the output. So DOM can be recycled, can be from your phytoplankton, in situ phytoplankton. However, it is also possible that you have, if you remember, your allochthonous source. Okay, allochthonous source. So not autochthonous source. So if you have allochthonous source, which means the bacteria varies independent of the chlorophyll because the bacteria is dependent upon an external source of DOM. So it's possible. And usually you may see this kind of phenomenon when looking at coastal water sites, coastal waters, on even estuaries. Why? Because coastal waters, lots of input from the land, lots of input, and estuaries, lots of input from the rivers, from anthropogenic activities. Okay, so that it's possible for uncoupling to occur. So, however, our observations of the determination of the marine microbial food web was based on observations where there is striking coupling between the different components. So this is kind of a study where you can analyze the data using correlation and correlation analysis. Then you can use regression analysis to look at the equation to link the two components. However, when we are studying water columns, so usually we work with contour plots. Okay. Okay. We look at contour plots where the y-axis will be the depth. So contour plots is actually like a XYZ plot, XYZ plot. Okay. Your y, if it's a water column, can be the depth of the water column. So it can be 50 meters, 100 meters. Okay. Then the x-axis is usually can be time, because this can be a time series sampling. So we can sample every month or every week over a couple 1 or 2 years. Okay, so we'll have points here. So these are all the samples that we have collected. Okay. And at each point, there will be a concentration. So your Z is the concentration of the parameter that you're measuring. So contour plots are drawn by a software. So usually they will look at the concentration of the plots of the points, and then they will plot out the maybe like this. Okay, so then from here maybe you can see the numbers here: 2, 4, 6, 8. So if this is a contour plot for chlorophyll A, that means here there is a high concentration of chlorophyll A that decreases over time. Okay, and after this phenomenon here, you only see the higher chlorophyll A at the surface area. Usually when we look at studies like this, you can see if the next study look at bacteria, you should expect to see a similar pattern, except that the bacteria will probably peak at a later time relative to the chlorophyll, because the bacteria is dependent upon primary production. So initial phytoplankton bloom here, the phytoplankton increase strikingly here. Then there will be DOM production, which will stimulate the bacteria later. And because bacteria is heterotrophic, their distribution covers a deeper area and not limited only to the surface. Then if you're looking at HNF, because you know that HNF is dependent on bacteria, you should see the peak in HNF occurring after bacteria. So this is how contour plots are used. when studying the water column over time, through time. Okay. So what contour plots are useful also when looking at estuaries. So this is something that most temperate regions will observe. You'll see a spring phytoplankton bloom. Okay, then you see a bacterial increase after the phytoplankton bloom, and then later protozoa increase. So from there, people began to accept the marine microbial food web. Okay, they began to accept it because the observations from the field fulfill the relationships between the different components. Earlier, I showed how the correlation analysis shows the coupling, okay, between the different components. Here, with contour plots, you can show succession. Okay, succession. Succession of the different components. So another way you can show this is probably by doing this. So this is chlorophyll A, this is bacteria, and this is their HNF. Okay. Another way you can show succession is by plotting this. So this is usually integrated column. So these charts are when you sum up all the concentrations at each time point. Okay, you sum up all the concentrations, and then you'll get a graph like this showing the succession from chlorophyll to bacteria to HNF. So we show that sometimes uncoupling occurs, okay, especially when there's an alternate source of food for the bacteria. Yeah. Usually when we talk about bacteria in the seas, we tend to focus on heterotrophs and not autotrophs. Although we know that there are autotrophic bacteria in the seas, their numbers are usually too small to play a big role in the food web. Okay, so we show how there can, from correlation analysis, you can show the linkage between the organisms. So another word for meta-analysis is also cross-system analysis. So this is what some of the scientists have done. So we look at the balance between primary production and respiration because of the global warming, the climate change problem, where we know that the main source of the problem is from the CO2 emissions. So scientists are more and more concerned about the balance between the autotrophy and heterotrophy, whether uh, whether a place will emit CO2 or absorb CO2. Okay, so if primary productivity is higher than community respiration at one place, at one habitat, so that habitat becomes net autotrophy. Okay, not net autotrophy means it sinks. It is a sink of CO2. CO2 is being, there's a net absorption of CO2 at that place. Okay, whereas if the community respiration is more than primary productivity, then it becomes net heterotrophy or source of CO2. So is this possible? Because when you think about the food web or you think about the food pyramid, the base is primary production. How can community respiration be more than primary production? So again, you must remember the source of organic matter. If it's only limited to primary production, then it's impossible. Community respiration will always be smaller than primary production. But the problem is humans have contributed a lot of organic matter, contributed a lot of pollutants to the seas. And that is why it is possible for community respiration to overwhelm primary productivity. So that is why most of the coastal waters is driven towards net heterotrophy because of the contribution from humans. In other words, the anthropogenic contribution. Okay, so in the tropics, in our waters, we are more concerned with climate change because our temperatures are already near optimum for most of the biological processes. So what happens when there can be two possible results from further increase in temperature? One is the mass extinction of biological organisms, and two, the increasing respiration of heterotrophs. Okay, because biological processes increase with temperature rise. Okay, so there will be more and more net heterotrophy when temperature rises. Okay, so this is the calculation of growth efficiency. We need to know sometimes the bacterial growth efficiency because measuring bacterial respiration is difficult and not everyone does it. So usually in some studies they will just assume a growth efficiency value of maybe about 20% to 30% and then estimate the bacterial respiration. So bacterial growth efficiency is estimated from uh, the ratio of bacterial production over bacterial consumption, where the bacterial consumption is the sum of bacterial production and respiration. Okay, so this is just a normal formula, the standard formula. So as with all components, there will be top-down and bottom-up controls. So top-down controls is defined by usually grazes or factors that cause mortality of the organism, whereas bottom-up controls usually affect the usually our food or other physical chemical uh, variables, for example temperature, that affects the production, the growth of the component. So we can have upper, middle, and lower estuaries. Of course, you have different types of estuaries depending on the geomorphology and the flow rates of the river. So if the rivers are fast flowing, you'll have different uh, shape in the geomorphology of the estuary as opposed to a slower flowing river. Of course, the geological structure of the rocks of the estuary will also determine what kind of formation is formed. So estuaries are very dynamic because it keeps changing. You have to remember an estuary is a place where the sea water and the river water interacts. And the interaction is not straightforward because you have to take into consideration the current, you have to take into consideration the tides. There are high tides, low tides, there are spring tides and neap tides. And also you have to take into consideration the rainfall. Okay, upstream of the river mouth, if it's rainy season, there will be more water going into the rivers, and the flow rate of the river will change, and that will of course affect the mixing rate, the type of mixing that occurs between the rivers and the seas. So a lot of variability. So imagine a place where things keep changing. Okay, so if the biological organism is not tough enough, cannot adapt to this kind of environment, they will not be able to survive well in a river mouth. So when we're talking about estuaries, we'll also think in terms of flushing rates and residence time. So flushing rates and residence time means how much time it takes before the water in the river mouth is being replaced. Okay, so that of course is dependent on the flow of the river and also the inflow of the seawater. So how long it takes before the water in the river mouth is being replaced or being flushed out. Okay, this one will have consequences because when you're looking at the pollution or any pollution studies, you are interested in the flushing rate because you want to know how long it takes before the pollution can be removed from the river mouth. You want to know that, okay? So yeah, because you want to make sure that the biological organisms there can survive. So if the pollutant remains in the estuary for a long time, it might have uh, irreversible effects upon the uh, biological community in the river mouth. Okay, so we have different types of estuaries. This is highly stratified because of the strong river discharge. So there is less mixing here. So essentially the fresh water flows on top of the seawater. So there's density difference here. So this is lighter. So it just floats on top like that with minimum seawater river interchange. There's a strong halocline. So halocline is based on salinity. So this is partially mixed, and this is uh, vertically homogeneous. This is when the sea strong turbulent mixing because of intense tidal flow. The sea is strong here, so quickly mixes up with the river water. So it's essentially homogeneous. Okay, and this is fjords, especially in the Baltics, where you can see a sill geological structure here that captures all seawater here. There's not much flushing here, and the residence time here is very long. This is usually anaerobic, so a lot of anaerobic studies have been done in fjords. So this is due to the melting of the glaciers. Okay, we can also have contour plots where okay where we look at the distance from river mouth. Okay, so we look at the distance from river mouth. So the x-axis is the distance from the river mouth, Then the y-axis is of course the depth. So as I mentioned, a contour plot is a x, y, z plot. So your z is the concentration of the variable that you're measuring. So if you're looking at so if this is the distance from the river mouth is 0, that means this is where the river mouth is. So this is the upstream. Okay. So if you were looking at an estuary where there's a strong halocline, then I would imagine if you are measuring salinity, okay, so this would be probably where you have mixing, so maybe about 15, 10, 5, or maybe 0. This shows the salinity. So here you still have seawater here, and here is where your fresh water is. So you can have contour plots that show the changes of the salinity with the distance from river mouth. Okay, so you can have contour plots that show distance based on distance from a place, or a contour plot based on time series. Okay, estuaries from the river to the sea. So from a calcium carbonate dominated river water to a sodium chloride dominated seawater, and where the concentration of salinity can be up to 30 parts per thousand. So the estuaries have steep gradients in ionic strength. Okay, so the main the main point here is when the water flows from the river to the sea, the ionic strength of the water changes tremendously. Okay, that causes a lot of colloidal material to flocculate and sink. That is why estuaries tend to have a filtering ability. It is able to clean the waters, where a lot of colloidal material will sink, and then you have clearer waters passing into the sea. So the estuaries can act as filters. So this is how you can use mixing graphs to see whether the component that you're studying is actually being removed from the estuary if you have a graph like this. So for mixing graphs, usually we'll have the y-axis where we measure the component, and an x-axis where is a conservative index of the conservative index showing the changes from the river to the sea. So usually we use salinity because it is a variable that is affected only by mixing. No biological processes affect the level of salinity. Okay, only by physical mixing. So basically, if you draw a x-axis based on salinity, if you have 0 here, that means this is fresh water. If you have 30 here, that means it's seawater. So from fresh water to seawater, if what you are plotting falls directly straight like this, that means the component you're measuring is non-reactive. It is just a theoretical dilution line. There is no addition or no removal from the water. So this is how mixing graphs can be used to check your ability of estuary to act as a filter towards the specific component that you are measuring.